So this is the issues in biotechnology, the way we work with life. The on-campus version here, and the online version listed as BCH 190. We're in section 1, covering the mechanics of DNA and general biotechnology. We've already investigated aspects such as setting the stage. We looked at the flow of life and understand now that life consists of information, and it's essentially can be described as an information processing system that's capable of replication with variation. And we looked at some of the tools in biotechnology. So we now know that we can clone pieces of DNA, and we can make expression cassettes called transgenes out of synthetic sections of DNA. And so tonight, in the first part, I want to go from how this information that we call a gene is translated into an organism. I'm going to attempt to describe how we go from information out to a living organism in about an hour or so. So how does gene expression relate to development? In the second part, we will look at more tools in biotechnology to investigate the genomics revolution. So first, gene expression and development. Do you believe in whales? It's not a rhetorical question. You know, going, gone. Marine mammals are rapidly becoming extinct. It, in fact, could happen in your lifetime. There used to be lots of whales off the shore here, in fact. Has anybody here ever seen one? I've seen 2 in the last 20 years. How about dinosaurs? Again, I don't mean this as a rhetorical question because, like I said, we see that 32% of this group believes in Bigfoot and about the same amount in the Loch Ness Monster. Overwhelmingly, yes. Some no. Some are maybes. We do, if you've ever been down to the Peabody Museum in New Haven at Yale, there's some great displays of some fossilized dinosaur bones. But, you know, I bring this up because of extinction. You know, that we still wonder about what happened to the dinosaurs about 65 million years ago. They, along with about 60% of the rest of the species on the planet, just disappeared. And that wasn't the first mass extinction. There's actually been several. If you go back a few 100 million years before that, a large number of the species just all of a sudden collapsed. We are right now in the period of time that has been called the Anthropocene. Yeah, the Anthropocene, the time of humans. In order to get named as a geological epoch, you have to realize that we've made a geological impact that will be detectable for the rest of time in many, many ways. And one of the hallmarks of the Anthropocene so far is that we are experiencing a rapid rate of extinction that is virtually unparalleled since the loss of the dinosaurs. But time has shown that species come and go. Maybe over 95, maybe as much as 99% of the species that have ever lived on this planet are not alive now. How do we ever know what we know? How do we know that dinosaurs did exist? Well, we have bones, we have some evidence. We could get really fussy about this, you know. And some philosophers have gotten really fussy about it, to the point where, you know, you can't prove the chair that you're sitting in. And this is, yes, that sounds absurd. Go to any of the existentialists for verification of this idea. But you could also talk about it in terms of biology, in fact. Yeah, you can't prove the chair you're sitting in any more than I can prove the existence of this thing, this podium. And the reason for that is that, you know, your perception is based on the information that you can take in, right? So it takes however many picoseconds for you to analyze that. You don't know what happened to you in that small fraction of time, right? You could evaporate, or the chair could evaporate. You don't know. By the time you proved it, it's already in the past. And the past is not a part of reality. It's a part of history. The future is not a part of reality either, because it hasn't happened yet. So the only reality really only exists at the cutting edge of the present moment, right? Make sense? So you can't prove the chair you're sitting in any more than I can prove a dinosaur. But given the evidence, I think most of you will concur that you're sitting in a chair. And I believe this podium exists based on the information afforded to me. So we can look at science that way. Certainly, nothing can be absolute on the premise that I just gave. However, the body of knowledge that you have based on your perceptions and the knowledge that has come to us through science over hundreds of years leads us to believe in what we call facts. We think that you're sitting in a chair. I mean, I know that sounds absurd, but really, this comes down to bear on what we think of in terms of evidence-based knowledge and observation. We are now more than knee-deep into this question, what is life? I've encouraged you to write your own 50-word definition. I will give extra credit for that. You saw mine last week. I'd be happy to make that one available to you. And we've quandered about how did life begin on Earth since the Earth cooled down? When did life begin on this planet? Could it occur elsewhere? What would be the necessary parameters for that to occur? And the best evidence indicates that life on Earth here started as single cells roughly about 3.2 billion years ago. So we realize that humans are in the last fraction of time. And we're not bright enough, you know, to get off of this solar system yet. We can barely get off of the planet. So it took 3.2 billion years of life to get to a point where you as a human being can listen to me talk about life, let alone work with it, or let alone get out of here. I mean, another galaxy is a long ways away. We'll get to this question next week. But the flow of information in the vast majority of life forms on Earth, we already understand goes from DNA to RNA to protein. So I want to start tonight with that premise. We've already laid the groundwork for this, that there is information in the chained molecule of DNA that's encoded in the sequence of A's, T's, G's, and C's that make up that sequence. And that can be decoded through an intermediate molecule of RNA by transferring that code directly and then decoding it into the language of proteins, where every 3 of those A's, T's, G's, and C's is going to spell for one amino acid. There are 20 different amino acids, and different proteins have different amino acid sequences, and therefore that determines their function. So a muscle protein like myosin has a different amino acid sequence and has a different function than, say, a liver protein like an alcohol dehydrogenase. So life is an information processing system capable of variation. Is that all? Remember, I had 50 more words to spend on mine, or at least 45 or so. And then you look at the connectedness of this, and it's awesome that, you know, here we are as humans, as just a small player in the bigger picture. And now we see in the Anthropocene what impact humans are having on the total situation, and it is indeed awesome. Some brief biochemistry before we get going into development, just to understand where we are with this. The flow of energy on Earth comes to life through sunlight, made by primary producers through the reactions in photosynthesis. So all chemical energy that life runs on comes to us through plants. The second tier of events are animals that eat plants, so-called herbivores, and then there are the carnivores that eat them, and the decomposers that virtually eat everything else, creating an entire cycle, an energy cycle. So light energy and energy transfer systems abound in both plants and animals for this reason. How do we capture the energy from the sun to make sugars is part of what photosynthetic reactions do. I'll get back into this in a later lecture when we talk about how humans might manipulate this for bioenergy resources. But plants have been making bioenergy for a long time, taking sunlight, using photons as energy to transfer that energy through an electron transport system and eventually split water. If we could do that, you know, energy-wise, it'd be game over. But I'll get to that later. Splitting water makes all the oxygen that animals breathe. There was not oxygen before there was photosynthesis. So the other half of that is the H2, and that creates a gradient inside of a chloroplast that drives a chemical gradient to make enough energy to take carbon dioxide out of the atmosphere and form a covalent bond with a 5-carbon sugar to make glucose. Therein, all the energy biologically on the Earth is captured. Mitochondria use oxygen to break down sugar in a chemical reaction called respiration. The currency, the chemical currency in all of those reactions is called ATP, adenosine triphosphate. 3 phosphates, and it's the covalent linkages between the phosphate bonds where energy is stored. So ATP is this very versatile player, and the energy is used for all types of reactions, including making chained molecules like DNA, RNA, and protein. That requires an energy input. Making muscles and muscle proteins contract is driven by ATP. Virtually most of the reactions that require energy are going to use phosphate bonds in breaking them, whether that's in ATP or GTP or like molecules. So enzymes catalyze reactions that would otherwise happen more slowly. A lot of the chemical reactions in life itself require enzymes. Enzymes are proteins. And as proteins, they were encoded for by genes. So all of the chemical reactions in life are mediated by enzymes. Enzymes are proteins encoded by genes. So if genes control the information to make those enzymes, they're really controlling all the biochemical reactions. If you have a mutation in a gene for a specific enzyme that's very important, you're out of luck, my friend. Especially if you only have 1 copy of that gene. I was harping on your hemoglobin gene the last time. You know, you don't have 1, and I'll get to this in the second part tonight. Actually, you have 5. In fact, you have 6. 1 doesn't work so well, I can tell by looking at you. But I'll get back to that story. Enzymes are proteins. And if you have a mutation in a gene, it is either going to make that protein work better or be benign, or it's going to mess something up. Most of the time, it's going to mess something up. So enzymes are usually proteins. Photosynthesis is arguably the most important reaction on Earth. And I don't say that because I am plant biased, even though I am. You can look this up, that, as I said, all the energy on Earth comes to us through photosynthesis. And if you're a biologist, oh, hell, it doesn't matter what you are. If you look at photosynthesis, you have to be amazed. If you look at how this works, capturing light energy is just a beautiful thing. It's a beautiful process. And I had mentioned this book, The Way Life Works, by Malin Hoagland and illustrated by Bert Dodson. You can look that up in terms of how they describe electrons in whirling dancers like they depict here in this chloroplast ballroom. But photosynthesis is also detailed in your textbook, and so you should look at that. But basically, by splitting water, a hydrogen ion gradient is built up inside of the chloroplast. And by having a large amount of hydrogen ions inside the chloroplast, these are funneled through a protein called ATPase, and that energy then is used to make ATP. The photosynthetic light harvesting complexes in plants are split into two main functions that have been recognized, photosystem 1 and photosystem 2. And I'll leave that description to a more advanced course. But I did want you to recognize this slide for, first of all, the complexity shown here in the number of proteins that are involved. It's not just that light is captured by plants and voila, you split water. You know, this is not exactly how it happens. Not at all. There's a complex of these proteins in this cluster that we call PS2, photosystem 2. And if you look at the top of that slide, there are light harvesting pigments, light harvesting complex pigments, chlorophyll A, B, and etc., that are capable of accepting light energy as a photon. This excites electrons in the outer orbitals of those molecules, and those excited electrons are then transferred down through this electron transport system shown here that's comprised of all these proteins that are stuck together in this particle, you see? So electrons are being transferred from one protein to another in this scheme. The energy is comprised in these electrons. So what you have to recognize here is that each one of those proteins is coded for by a gene, right? And this thing is pretty complicated. Down at the middle of that, you see 4 manganese atoms. It's at that point where water is split. There are scientists all over the world right now studying exactly what happens right there, because right there is where water is split. I'll make a case for this later. Humans should learn how to do this. If we do this, we're out of carbon. We are out of a carbon energy equation. If humans learn how to split water, you don't need oil. You don't need gas. You don't need nuclear. You don't need wave or wind. You split water, you have an unlimited source of energy. And when you burn hydrogen, you get a big bang, right? The outcome of that big bang is pure water. So what do you do by making photosynthesis that would be artificial by humans? If you did this, if you were capable of making this artificially, right, you could split water. And by splitting water, you would solve the oil crisis. You would solve the energy crisis and all the wars that will happen when people struggle over the last few drops. You'll solve the climate change crisis because you're not using CO2. You're not making CO2. You're not using carbon. You're not using fossil fuels. You'll solve the impending water crisis because you use water and make what? Pure water. I'm preempting a future lecture, but I would say that's the future. And I don't know what you're planning on doing with your life, but I hope it involves this. I think we all ought to get off our butts and do this, really. I personally think there's not another important thing to do, but I'm a little biased on that. Respiration takes the energy from photosynthesis, the energy that's been captured in sugars, and turns those sugars into energy, liberating CO2. So you can see the whole cycle comes full circle right there. So the basic idea here is to use sugars and oxygen to, again, make that currency of ATP by making a hydrogen ion gradient. So respiration turns sugar into energy and it liberates CO2. So realize that all of the enzymes that are involved in these reactions are proteins that are encoded for by genes. There are various diseases in humans and other organisms that are caused by just slight mutations in those proteins. Every single protein in that scheme is important. So the flow of energy goes from sunlight to plants to animals to fungi and bacteria, and a whole cycle of carbon is created. From physics, we understand the first two laws of thermodynamics, the first one being that energy can be gained or lost, cannot be gained or lost, it's just shifted from one form or another. So it's never created or destroyed. The second law predicts that everything that is in order tends to disperse. I'm sure if you've ever seen my car, you would realize that that's the case. Or my office, you know, there's a tendency to disorder. Maybe your room is the same way. This is called entropy. So there is a general tendency towards disorder and entropy. So as energy leaves a situation, molecules become more disorganized. So you go from a solid by applying heat energy to a liquid. Think of ice going to liquid water. More heat goes to a gaseous state. So you're going from order in a crystal water structure to a liquid structure to a gaseous disordered structure. Some people have said that this second law of thermodynamics threatens an explanation of how complexity has increased over time with biological life on Earth. How could this possibly be? Instead, actually, it guarantees it because actually there's a steady stream of energy coming in from the sun. And it's that energy from the sun that is driving the force against entropy and disorder. So the energy from the sun is accumulating and stable molecules are built with that energy to assemble information and to change. So actually, the complexity of life that we see on the planet is very consistent with the laws of thermodynamics. Okay, I said that now that we understand that we have biochemical pathways, and again, I would invite you to my office sometime. I have this exquisite map of all the biochemical reactions on my wall. It's amazing, really. I mean, if you think about that, and first of all, for humans to even know that is amazing. But when you look at the complexity of all of the reactions that it takes to run a machine like this, it is really awesome. So now in this part, I wanted to describe how that system is actually regulated. Given the fact that this thing is so complicated, How do you go from a rather homogeneous egg out to something that's sitting here in a chair and looking at me? At least I think you're sitting in a chair. Wait, we got over that. So this is really about gene expression and development. How do we go from a segment of DNA, or 24,567 of them, out to a human organism? Or for that matter, a rice plant. How is that regulated? You can't just simply turn on 24,567 genes and end up with a human being. You'd just be a sack of protein. And that's not really that interesting. So this reading assignment for this part of the course should involve Chapter 5 on DNA gene expression, and you might want to also look at those applications in biotechnology. Life is diverse. Life is diverse. The many different plans. But when you start looking closely, remember I said in the last time about emergent patterns? And if you look at it, you think, my gosh, how could this possibly be, right? I mean, it looks very complicated. It is very complicated, but it's not as complicated as all of those individuals seem to make it seem. Remember I gave you the example of a fractal. When you look at a fractal, those images that look very complex actually are created by the reiteration of a simple algorithm. Similarly, we will see genetic patterns in organisms that are reiterated with some variation. So what seems very complicated at the outset has actually a simple explanation, or at least a more simple explanation than it would seem from the diversity that you see. So genes are segments of DNA that they code for everything? Really? Do they code for lipids, sugars? They code for proteins. So, you might want to reconsider this. Genes code for proteins that make all of the other stuff in living organisms. Genes code for proteins. Proteins run biochemical reactions that make all the other stuff. But by controlling proteins, you control what lipids you make, how fast you digest alcohol, and virtually anything else. So genes code for proteins, and some of these proteins act as switches that turns genes on and off. So how one gene can turn another gene on and off. The basic idea is that you have all of these different genes that have to be regulated. Some of them will be regulated by signals that they receive from the outside. Could be hormones, could be an environmental signal. So the basic process is a gene is going to be decoded first by a process called transcription, where the code for a protein is going to be transferred from DNA onto RNA, leave the nucleus, and be decoded by a ribosome in the cytoplasm, where every 3 bases is going to code for a separate amino acid that will build a chain of amino acids that is specific to a certain protein. Remember my description of prokaryotes and eukaryotes. Prokaryotes being bacteria, eukaryotes, plants, animals, and fungi. And in eukaryotes, I already showed you that a typical gene might be made up of a coding sequence. In eukaryotes, there's another interesting factoid, that the gene sequence for a protein isn't just from start to finish always, but they're broken into these 2 different segments called introns and exons. Exons are the coding region for the protein. Introns are intervening sequences that are clipped out before it leaves the nucleus. The reason why this is important turns out to be in evolution, as we'll describe later. But your 24,567 genes, you make more than over 100,000 proteins. How is this possible out of that information? We can shuffle these exons around. One gene might code for more than one protein. That's how that's done, and that's why I mention it here. So the translation process in eukaryotes means that those introns are excised out. They also lend stability to the messenger RNA. So your 3 billion base pairs of DNA in a human, only about 4% of that codes for genes, probably less than that. What's the rest of it doing? 96% of it is not coding for protein. There's a lot of non-coding DNA around. There are regions of DNA that don't code for protein. You may have seen in the news, actually just about a month ago, I don't know if you read these things or not, but the non-coding DNA had for a long time been referred to as junk DNA, and there was a program that was 500 scientists collaboration that really looked at decoding what the non-coding DNA does in humans. Junk no more. That's about the way we could put that. So we're getting an understanding about what the non-coding DNA does. And I'll get back to that in just a minute. So gene expression, how genes are decoded into proteins and how that is regulated, tells the story about how we go from a homogeneous egg that once fertilized becomes polarized. That's the first thing that happens in nearly everything, a top and a bottom. Once that process starts, everything becomes differentiated after that, until one cell becomes 2, becomes 4, becomes 8, becomes a hollow ball of cells if you're becoming a mammal, and then eventually an embryo. How does this actually orchestrated? How do we turn genes on and off in order to control whether or not you have an epidermis, an ectodermis, or a mesodermis? How do we get these different organs that all have these different functions with different biochemical pathways in them? We also know that every time you copy your DNA, mistakes are made. A's go in sometimes where T's can go in. This could be the basis of some diseases if they happen in body cells that we call somatic cells, but they can become heritable changes if they happen in your gametes. And this gives rise to the variation that I see here. But it's not just A's and T's changing places. We can have all kinds of things take place. I mentioned in the last lecture that during the making of gametes, whole chromosome arms from your mom and your dad can swap places. This is not always a precise excision insertion event. Let's imagine you have exons that are from your mom and your dad that behave differently. Now in that process, they get swapped out. You've got an entirely different gene now. That's variation. That's just one of the mechanisms. I want to impress upon you that this whole process creates enough variation to generate not only what I see in this room, but what I see in the entire world of life. We don't need to argue about whether or not there's enough variation out there to create life, okay? And we don't need to argue anymore really about the mechanisms that drive that. Given DNA, given information like what we have, there's plenty of ways to make variation. And if you put a filter on that, and the filter being what works versus what doesn't, you will end up with exactly what you see outside. I'll make a point to that later. So how do genes work? Here's an overview that's from your book in chapter 5, where inside the nucleus, the piece of DNA that we call a gene is transcribed into RNA. and then that leaves the nucleus as a messenger RNA and is translated into proteins. Okay, let's try this one. We'll talk about how environment and genetics play a role in the outcome, which we call phenotype. Phenotype is the outcome of variation in genetics by variation in environment. And I'll make a point to bring that up in a little while. So we already know DNA goes to RNA goes to protein. So since all of the genes that code for all of the proteins are in an organism are present in a cell, how is development controlled? I should make a point here that prokaryotes, however simple they seem, have an internal development of their own. This has recently come to light by investigations that proteins inside of even prokaryotic cells have certain spaces that they go to. But how is a gene turned on and off was first examined, or first detailed really, by the French geneticist and biochemist Jacob and Monod. And they were looking at E. coli, okay, common gut bacteria in mammals. You may have heard about it because of its pathogenic strains that sometimes cause people to get sick. But E. coli, Escherichia coli, is the genus species of this bacteria, is characterized by the fact that it's in mammals and some other animals, but it can digest milk sugar. So that's what it's doing in the intestines of mammals. And Jacob and Monod were looking at how genes that are involved in making enzymes that digest milk sugar in E. coli are regulated. What they noticed is that when there is no lactose around, the genes that code for the enzymes aren't making their RNA and aren't making their protein. So when there's no lactose around, it's not like these things are being wasteful and willy-nilly just making the enzymes to degrade them. Rather, what they discovered through investigations of mutants, basically, was that there was a gene that they called a regulator. The regulator codes for this protein that's called a repressor. This repressor, when there is no lactose around, binds to an upstream segment of the DNA called an operator. So in front of the sequences that are coding for the enzymes, there's a sequence that's a switch that's going to regulate whether or not they're going to make these enzymes to degrade lactose or not. If there's no lactose around, this protein called a repressor comes in and sits there and stops RNA polymerase from reading those genes, as shown in the middle part of this slide as that red circle. So RNA, when there's no lactose around, there's no messenger RNA coming off of those genes to make the enzymes that will degrade lactose. When lactose is around, as depicted in the square binding to the repressor in the lower right side of the slide, it changes the shape of that protein. So the protein then comes off of the promoter, off of that operator part of the promoter, and makes that region now accessible to RNA polymerase. You get the RNA coming off, and it's decoded into the enzymes that then degrade the lactose into energy for the bacteria. When they've degraded all the lactose, that repressor goes, sits back down, and shuts off the system. You got it? This is a very important example, because basically this is how everything works. It's when there are proteins that can recognize certain DNA sequences. This is an important fact too. And this is going to come up all over the place. This one happens to be called a repressor. It binds to the protein, or binds to the genes in front of the promoter, in front of the genes for lactose metabolism, shutting them off. When there's lactose around, it changes the shape of that protein, it comes off of the DNA, and we read it. So it's a whole feedback cycle. But the promoter is a segment of DNA that doesn't code for a protein. It's a switch that turns genes on and off. So you can imagine that you have eye-specific promoters making eye-specific proteins. You have liver-specific promoters making liver-specific proteins. All of those biochemical cycles that I just showed you and all of those enzymes are regulated just this way. So your 24,000 genes or so. In this chart, you can see that hemoglobin, for example, as a blood protein, is on, it's active, it's making it, when I say the gene is on, it's making its RNA and making its protein. It's on in red blood cells, but it's off in egg, off in skin cells, off in muscle. Keratin is a skin protein, a structural protein. It's off in eggs, off in red blood cells, and on in skin. And a contractile protein like myosin is off in egg and red blood cells, but on in muscle cells. So these genetic switches in front of genes are called promoters. So the expression of a gene transcription is then the transfer of information from DNA to RNA, as depicted here as the answer B. So this regulation of gene expression and turning promoters by using promoters to turn genes on and off. Here's another example about tryptophan, that's an amino acid that is made by some cells, and in this example by bacteria, where a repressor, when there's tryptophan around, you don't want to be wasteful and be making it. So tryptophan binds to its repressor. And again, depicted here, the RNA polymerase does not read those genes to make tryptophan. But when tryptophan is sparse, the tryptophan pops out, the repressor falls off, RNA polymerase comes in, separates the DNA strands, makes the RNA, which is decoded by the ribosome and makes these proteins that then start making tryptophan again, and when tryptophan becomes abundant, it binds back onto that repressor and shuts the system back down. So promoters are switches that regulate gene expression. So you've seen this diagram before in another fashion when I described how we clone DNA, and I mentioned that we can make synthetic genes by cloning a promoter from, that would regulate gene expression onto any coding sequence from any source. But this also happens to be the basic structure of a gene, where you'd have a promoter that is upstream of the coding region of the gene, driving the expression and determining cell specificity. The start of transcription, typically recognized as what's called a Tata box, T-A-T-A. Named after Goldberg-Hodgeness. It used to be called the Goldberg-Hodgeness box, but now it's called a Tata box, and that's kind of a consensus sequence for the start of transcription by RNA polymerase in a promoter. And these promoters are regulated by proteins that will recognize certain sequences on the promoter and regulate their expression, whether that is cell-specific or metabolic-specific or developmentally specific. Downstream, there is a termination signal that will stop transcription, and usually in many organisms, the termination signal is capped with a polyadenylation signal, AAAAA, to kind of lend stability to that messenger RNA. But here's the diagram I showed you of a transgene, which can be made to more or less mimic that same structure of a gene, where we can cut and paste promoters from any source next to coding sequences from any source, and then follow it with a termination signal. So A transgene in biotechnology then is a synthetic gene that will follow the same exact expression characteristics and rules that we see in genes in organisms. Let's just make sure we're on the same page here. Yes, it's a promoter. A termination signal is at the other end. It's definitely not a Nixon. So just to be clear, again, these upstream segments which have DNA binding sites where different proteins, such as the example I showed you in E. coli, as a repressor, can bind and activate this. They're not always shutting genes off, but also activating genes. And the so-called TATA box is usually 30 base pairs or so upstream from the initiating ATG methionine encoding codon that starts most proteins. And so this diagram shows you that you can have different proteins that will recognize sites in a promoter. A promoter can exist for about 1,500 base pairs upstream of an actual gene. The gene, in fact, might be a smaller sequence than the promoter that's regulating it. So we can code for genes, we can clone promoters, we can clone genes. But in amongst that promoting region, that promoter region, there will be specific proteins that can recognize specific sequences and bind there. And so it's this concert of binding proteins. The simple example was the lactose operon. But in regulation in general, there might be a concert of these different DNA binding proteins that regulate gene expression. And here's another example. I put a couple of these in here, but this is how RNA polymerase might be diagrammed in recognizing the start of transcription after it's activating. And we can sequence these and compare promoters from one organism into another. And I'll get into this in the second part of the lecture tonight on genomics. So if we're interested in expressing a gene in another organism, as we are in biotechnology approaches, we might want to compare those promoter regions and those functions. And we can look at how promoters for different genes compare in different organisms, as shown here in this comparison between the relation between different primates, humans most closely related in this case to chimps, and the other primates shown here in this clade diagram. So these so-called DNA binding proteins that recognize sites on promoters are collectively can be called transcription factors. So transcription factors themselves are proteins. So what do you have here? It's like a chicken and the egg thing, really, where you've got a gene that's coding for a protein called a transcription factor that is going to be regulating other genes. So you've got a gene making a protein, and that protein's going to regulate a whole bunch of other genes. Follow that? So you've got one gene that's going to throw like a master switch and say, oh, we're going to make this protein that's going to recognize all I proteins. Throw the switch and all I proteins come on. And if you have a mutation in one of those, you're pretty messed up. Or different. In 1969, Roy Britton and Eric Davidson wrote an article called Coordinate Expression of Batteries of Genes. And when they did this, people kind of thought they were nuts. But the title of this paper pretty much explains the situation. Coordinate expression of batteries of genes. In other words, a transcription factor that's capable of regulating a whole set of genes. You throw that switch and the whole set is regulated. We don't have to do it willy-nilly or just one at a time specifically. We're certainly not doing it uncontrolled. So they hypothesized that it would be this coordinate expression of batteries of genes. You throw this switch and everything comes on. And in fact, they were pretty close in their description of how this thing had to look like, and this has been borne out. Here's a paper that I pulled out of Genes in Development that was accepted just this year in April on coordinate regulation of neural mRNA steady state levels through developmentally controlled intron retention. So these shows you how batteries of genes are controlled in neuronal development and roughly substantiates what Britton and Davidson thought. So you have a cell, and it's got all these different proteins in it and these different compartments. And if you took the proteins from the different compartments out of a cell and organized them as such, you could see that there are different levels of these different proteins. I think that's a pretty neat diagram, actually. But how do we do this? Well, one example of a master switch gene, as diagrammed in this slide here, is a gene that's referred to as the Hox gene set, which is universal in animals, or seems to be universal in animals. And it's a transcription factor that is regulating segmentation during embryo development. So you can see that these different switches shown here in the fruit fly, down in the lower left, the mouse in the middle, and the human to the lower right, all have a lot to do with each other and are all very similar. So where red is red, for example, it's red in all three organisms. These are color-coded depictions of Hox genes that are controlling development that's involved in these different segmentations. And we can see when we look at the sequence of Hox genes that there is a strong homology between these different organisms, such as fruit fly and human, that control segmentation. So these switches, in fact, build on switches. And as I mentioned, if you have a mutation in a major transcription factor gene, it really disrupts all downstream development that's associated with controlling the coordinate expression of those batteries of genes. And this has been well studied in organisms like fruit fly, Drosophila. But it's also, you can see examples as shown here in these different mutations in different fruits and vegetables. This is out of your textbook in chapter 5, but mutations or major switch genes cause this cascade effect. And here's an example also in your textbook of normal fruit fly eye development, which has been deregulated in antennae, and so the eyes then in this mutation are forming where antennae normally should. We can also cause disruption in transcription factors that are involved in development of certain diseases by field effects. One might be by exposure to tobacco smoke, as shown here, or sunlight, or by radiation, can cause mutations in genes that affect the regulation of cell division, for example, that's involved in tumor growth. But mutations can also be involved in leading to other illness. So right now, looking at the sequence of genes and their promoters is becoming an important factor in looking at controlling health and studying health in certain diseases. We just saw a compendium published just this week on the genes that are involved in regulating breast cancer, for example. This has been a long time coming. So these master genes that control the expression of other genes code for, let's be clear, yes, thank you, proteins for the most part. You guys, go back and read up on that. So again, this basic idea is some signal be it sugar, be it light, be it a hormone, is going to interact with some receptor that is going to send a secondary messenger protein that's going to act with transcription factors that are going to regulate a battery of genes. So remember my diagram of this bacteria. Every single color that you see there is a different protein encoded for by a different gene. Look at the intense regulation that must occur with those genes just in this single prokaryotic cell. Now think of this. The networks of genes and molecular maps and genetic circuits that are involved in maybe what's called networkomics now create these maps of how different genes and different gene products are involved in their regulatory pathways. So again, how do we go from an egg, once it's fertilized, to start dividing, to becoming organized into a blastocyst, shown here with its inner cell mass that will become the embryo proper, and its outer cell mass, which will become the extra embryonic tissues like the placenta, to eventually, through cell division, change planes in development to become organized into different tissue layers, and then into a fully formed organism and into a human embryo. Or if you look in this case at a cross-section of an eyeball, one thing you have to say is beautiful, even if you don't know what's going on there. That is just beautiful. The journal Science, by the way, holds a challenge every year to submit the most provocative images, and I always look forward to that issue. It covers all different kinds. This was one of the winners last year, showing the different patterns in gene expression associated with the developing eye, or in this case, trichomes on the surface of a leaf. These are hairs on the surface of a leaf. This is a hotly debated topic, but I'm just interested in your point of view. When does life begin as a human? That is provocative, since people vary on their opinions about this. States vary on their opinions about this. Even the Supreme Court varies sometimes on their opinion about this. Yeah, you know, that's common. That's common. That's most common. B would be the most common answer. At birth. You know, so you can see in this class how widely the answers differ. So there is no right answer to that question, really, except I would say this. We're in a biology class. By any of the criteria I have put forth so far, eggs and sperm are alive. There you have it. When? What's that? Well, that's what a lot of states and a lot of governments and a lot of people would say. I say there's no right answer to this, except we would say that by any criteria, a biologist would draw that an egg and a sperm are alive, right? Okay. That's just what we would say. We could argue about this, and now, like I say, the Supreme Court argues about this, so I don't think that here in BCH 190 we're going to come up with a definitive answer here. But this biologist would recognize an egg and a sperm as a living cell. Is that okay with you so far? Well, we could also get into how this might figure into future legislations on the topic, but I think that is certainly beyond the scope of this class. So how do even simple organisms like dictyostelium, which is a slime mold, form complex structures? How does an embryo develop? As depicted here, it's as if one cell were like a family that builds a house in the middle of nowhere. Later, using the same set of plans, the family's grown children build next door, and it's like like one egg becoming two cells. At the same time, using the same plans, eventually we have a larger grouping that becomes a cluster, and then by combining different parts of the building plans, that is like different genes being turned on in different parts of that organism, some builders create new kinds of houses. And during human development and other animals, and even in plants, cells move around. So these modified designs inspire their neighbors who copy them, and some owners move their houses nearer to the center of the community, and from this, one cell becomes many. And I've already brought up the example of your face, how the fabric changes but not the pattern. This brings me to my two cats. I found these two cats in a dumpster, and I named one Evolution. Then I found this other one that I named Development, and I noticed that sometimes they got along, and sometimes they just didn't. And that's the story of Evo Devo. So actually, Evo Devo is a whole field of biology now, not studied by these cats, but rather a group of scientists who study the influence of development on evolution and the influence of evolution on development. Realize that if you have a change or a mutation in a transcription factor that alters development, you can radically change an organism. Take a human face. It's flat in comparison to other primates, which is conical, you know? So don't you think that the architecture of the human head had something to do with a flat face? For sure. This is regulated by coordinate expression of batteries and genes, which is regulated primarily by a very small number of transcription factors. Therefore, a mutation in a single transcription factor could have a large impact on cranial development. Why is that important? A simple flat face allows for a bigger head with a bigger brain. And all of a sudden, things can change rapidly. So we can talk about evolution happening in slow mutations that occur over time. But you can also talk about what Stephen Jay Gould called punctuated equilibrium. Very rapid changes happening which have profound effects. We can also see that certain patterns in development are maintained. Much has been made, for example, out of, say, the Hox genes that are parallel in many different organisms controlling segmentation. But also a lot has been made of diagrams like this, where many different embryos from many different sources go through similar stages of development, perhaps reiterating earlier stages in evolution, and a lot is made about that observation. However, really what does it reflect? That common ancestrally has similar genetic patterns that these things are experiencing. That's really saying the same thing, just a little bit differently. So for example, if fish evolutionarily came before mammals, we see that early in development, all of these different embryos share gill slits early on, even up to this stage. This has caused some to reflect upon this characteristic that ontogeny recapitulates phylogeny. And like my previous statement about teleology and phylogeny, ontogeny recapitulates phylogeny. Again, if you say that at a dinner party, people will probably move away. But let's figure this out. Ontogeny is the origin and development of an individual organism. To recapitulate, to redo, to reiterate. Phylogeny is the evolutionary development. So the repetition of pattern. This is a repetition of pattern. It's consistent in all those different embryos and all those different organisms. You see? They have something in common. That is their genetics. The reiteration of pattern in life. And we see this kind of homology all over the place, whether it's pigment molecules like chlorophyll and hemoglobin, which are exactly the same, except one has a manganese cluster and the other one has an iron. Or chemotaxis that I brought up before. If you put a motile bacteria on a Petri plate, and acid over here and sugar over there, this motile bacteria will swim towards the sugar, sensing it chemotactically, just the same way white blood cells will swim to a site of inflammation, owing to a signal. And isn't that the same as consciousness? Really? Or an extrapolation of that would be free will, wouldn't it? Information. From information to an organism. And that brings us to cloning. All the information in an organism, in a eukaryote, is in its nucleus, in its DNA. Animals can now be cloned. Sheep, cattle, pigs, mice, rats, cats, dogs, mules, frogs, and now even primates. And we can engineer them. We can take all of the DNA in a separate nucleus, as shown here in the upper right, and inject it with a small micropipetting needle into an egg that's had its DNA removed, held here by a micropipette with a slight suction off to the left, and then injecting it, as shown here in the lower right. Stimulating it with an electrical impulse causes those cells to start to divide. That embryo then, with that new nucleus in it, is implanted into a surrogate mother to give rise to an offspring that was the same as from the donor cell that it came from. Shown here is what might be depicted for the first cloned sheep, Dolly. And this is from your textbook towards the end on cloning. So cloning then is a process where all members are directly descended asexually from a single organism by various ways. The clone that your grandmother makes of her African violet on the windowsill is nonetheless an asexually reproduced vegetative structure. It's, complicated anyway, even though it's just on your grandmother's windowsill. Still, we can take all of the DNA from a mammalian cell in a nucleus, as I've shown, and this demonstrates that DNA provides the instructions for an entire organism. So we can clone cats. This is the first cloned cat called Cece, for copycat, made by an organization called the Savings and Clone. That was a real company that offered services to clone your pet. I think they're out of business now, though. But they did clone the first cat. Mules were cloned by the University of Idaho when I think they were exceptionally bored. Horses have been cloned. Pigs have been cloned. In fact, clones of clones of clones and clones of pigs have been made. That's a later story when I talk about medical biotechnology. This is Dr. Jerry Yang, who unfortunately passed away a few years ago and lectured in this class. I was quite honored that he got up. He was a friend of mine. And he got up and he said that he was pleased and honored to speak to this class, and that this was his first speaking engagement since his last brain surgery. But Dr. Yang was the first person to clone cattle ever in the world and develop, gives me chills, really. You can go to Yukon and still see Dr. Yang's work over on Horse Barn Hill, where he was the first person to clone cattle, and he also developed some very innovative technology for in vitro fertilization in cattle and moving cattle into developing countries. One thing I want you to recognize by these clones here is that they're not all identical. I'll get back to that in another lecture, but that's also transcription factor regulated, and it's very similar to identical twins. Anybody here know identical twins? I have 2 nephews. Yeah, there's always something about them that's different. You ever notice that? Like even my 2 nephews, you know, like even their personalities are somewhat different. One's really cool and one's not. You know, I hope he doesn't see this. But humans will be cloned. The technology's on the shelf. To my knowledge, they have not been. Primates will be. The first steps have been taken already in monkeys. And couldn't we genetically engineer them? In a later lecture, I will position possible reasons why we might even do that. Would you clone yourself if you could? No, huh? No. As I said, there might be legitimate reasons to do this from a medical standpoint, and we'll get into some of those when we talk about ethics later.